going to talk about today is a new hypothesis that Professor David Deemer and myself at UC Santa Cruz have formulated over the last decade. And it was published first in 2015, and the full hypothesis was published in Astrobiology Journal last year. And we call it the hot spring hypothesis for an origin of life. It's one of several in our field in origin of life prebiotic chemistry. And let's start with a very interesting quote. And can everyone, by the way, check in, can everyone see this? Elias, you see this?
Yes.
Yes, it's okay.
We're good. Okay. 150 years ago this year, and this hasn't really been noticed by many of us, but I'm going to let you know about it today. Charles Darwin wrote a letter to his friend T.J. Hooker, and everyone knows the first part of the expression of this sentence in his letter. But if, and oh, what a big if we could conceive in some warm little pond somewhere with all sorts of ammonia and phosphoric salts, light, heat, et cetera, present, that life could begin. So this was his statement or his intuition on where he felt life could begin. But this second part of the sentence has come to light in the 21st century. And I'll read that for you. That a protein compound was chemically formed, ready to undergo still more complex changes. And this is a tremendously insightful statement, because what Darwin is saying is a protein, which Victorian science didn't know that it was a chain of amino acids, but they did know proteins existed, would form, i.e. a polymer would form and become more complex, become able to undergo more changes, which might mean that it might become longer. It might have become functional in some way, become more complex. And this is where we're at. In the origin of life, we have to find ways to grow polymers in our prebiotic chemistry. So I'm going to start right off with the full hypothesis for everyone. And then we're going to take it all apart experimentally and theoretically. There it is, the hot spring hypothesis for life. And what we're seeing here is a volcanic landmass on the early Earth 4 billion years ago. And the glowing thing with the disk up there is the sun with a very dusty, icy disk of planets forming. Well, they have formed by 4 billion years ago, but there's a lot of material. And you can see perhaps Venus there is still vacuuming up material. And so a lot of infall is happening, a lot of dust and meteorites, interplanetary dust particles are coming into the Earth's atmosphere. And the atmosphere is perhaps brown, perhaps very hazy, and this volcanic island is very active. And what we see is a caldera there with a hot spring. There would have been hydrothermal fields on the early Earth at this time. So a lot of the synthesis of organic compounds happens in that disk. It happens in effectively what you could call cosmochemistry or astrochemistry. Material then falls into the atmosphere and can accumulate on land in little pools. But in the bulk of the ocean, the global ocean would have been at that time, would just simply be diluted and lost to chemistry, to chemical reactions. So by accumulating on land, it can become concentrated. And that's a key for any chemistry to work. To get things together, you have to have concentration. And so after the accumulation of all of these compounds, they can form structures. And some of the structures are membranes. And other structures, as we'll see, are polymers, the two basic components of what we'll call protocells, the precursors to living cells, the deep precursors. What we'll find then, as we predict, is that this material can accumulate and find its way into cycling environments. And you'll notice that in Darwin's expression, he talked about undergo further complex changes. Changes is key. You need to have cycles to push basically push molecular systems away from equilibrium. So they're not simply just running out of steam, they're becoming more complex. And we think that this happens in a pond or a small pool subject to wet and dry cycling. So constant filling and refilling. It could be the accumulation of precipitation, dew, things like this. But geysers are so periodic, so regular that they can form an engine that can do periodic cycling in these pools. And we can see that all over the world in places like Yellowstone and in Rotorua in New Zealand. So we think that this is the engine that can lift systems away from equilibrium, drive the overall chemistry so that life emerges and then distributes across a landscape, a fitness landscape, if you will, down into different watery settings like a lakeshore, like streams, like wet environments within and underneath rocks, this sort of endolithic lifestyle. Life gets more robust across such a landscape and is adapted to, say, for instance, the saline estuaries and seashores. And once you get adaptation to a salty environment, which takes quite a bit of technology, and that you can have global distribution, at least at the marine margins. But life would start in a freshwater setting instead of in the oceans, and then adapt to the ocean environment. And this is borne out to some extent in the rock record, because if you see on the left side, you see these 3 rock samples. The first is geyserite, discovered about 6 years ago in the Pilbara in Northwest Australia, which shows evidence of life thriving in hot springs 3.5 billion years ago. And the next little rock sample is lacustrine, or lakeshore stromatolite, evidence of life at a lakeshore 2.7 billion years ago. And the bottom sample shows a marine stromatolite billions of years ago. So the rock record actually does show land as far back as we can look, and in a hot spring setting as well as other freshwater settings. So let's take a slice out of this land and look at what we mean by a fitness landscape. So for example, we believe that the Goldilocks chemistry, the ideal chemistry for starting life, is in a freshwater environment. It's heated, it's cycling, it can be fed by atmospheric contributions, by meteoritic contributions, and that life begins, adapts across the landscape to more extreme settings, and eventually can colonize the shallow marine, and it finds its way into the deep marine hydrothermal vent environment where it has been thought for decades that life could start. We think that that's the place life ends up, but can't begin there. And because the land has all these facilities, it can collect material, concentrate it, cycle it, and then adapt it and stress it to subject early life to stresses to make it more robust. It's effectively a physical fitness landscape with gradients. So all of these adaptations, phototrophy, protein translation, pores, all those things can arise in this environment. So just quickly showing you this rock evidence. These are different samples that with our colleagues at University of New South Wales have shown us on field trips. And you can see living stromatolites today at Shark Bay in Western Australia. So this is just a fascinating aspect of this work that geologists are working now deeply in the origin of life. This is a beautiful article published last year about that all the elements necessary for life to begin have been found in samples, in rock samples, in this very, very ancient Paleoarchean crust. So really, in terms of the soup of life, all the ingredients have been identified as available. Some of the other key ingredients are the organics. So amino acids, carboxylic acids, forming membranes, identified by Dave Deemer and colleagues 30 years ago in the Murchison meteorite. So they ground up portions of this carbonaceous chondrite and it formed membranes in solutions. So then there's other experiments. The recent mission to the comet by the European Space Agency found that 40% of the organic compounds in the dust surrounding the comet, 40% of that content is organic compounds. So what is the process that makes all these organics in space? Well, there's a number of excellent papers on this subject, and you can sort of see a little bit of a summary of how all of these organics, amino acids, carboxylic acids, nucleobases, sugars, and things like that can be made in space. And that there's sort of a chemistry cycle which would deliver these organics to the surface of planets, of terrestrial worlds. And that these feedstocks can concentrate on land but are lost in the dilute bulk of ocean. So an ocean world couldn't support this sort of cosmo-sourced pre-organic or prebiotic chemistry. So zooming forward a little bit here, this is a device that Dave Deemer built at UC Santa Cruz about a decade ago, which created an artificial hot spring. So you could hydrate little vials in a rotating plate and then subject them to dehydration through CO2 gas. And in this device, he placed mixtures of the building blocks of RNA, AMP and UMP, for example. And one of the things that happens when you dry down the solutions, if you have the presence of lipid, actually, even if there isn't lipid, but lipid amplifies this effect, water evaporates, water becomes a leaving group and pulls an ester bond reaction forward and creates a polymer out of, say, for instance, adenosine monophosphate or AMP. And RNA-like polymers can be produced easily without enzymes, without the enzymes of life, in a few cycles of 4 to 24 hours. And here's some little gel evidence and evidence from the nanopore sequencing device that Dave invented in his laboratory years ago. It's now a commercial product. So this is a way through wet-dry cycling, you can produce, we're talking 40 to 150 mer polymers of RNA and RNA compounds. Other groups, that notably the Nick Hudd's Center for Chemical Evolution, has used wet-dry cycling to produce peptides from amino acids. Recently, work has reproduced this at ELSI in Japan, and Dave is also self-assembled DNA. And let's take a look at this. So this is a gel scan of radioactive oligomers, which what Dave did was produce oligo T, so one of the bases of DNA, self-assembling and wet-dry cycling, and then ligated it to a known sequence of DNA and ran it through the nanopore sequencer. And you can see it here on the right, the nanopore sequencing output, and you can see all the T's. So that's kind of a shot heard around the world, and that DNA can also self-assemble under these conditions. So what we have is the self-assembly, and this is a AFM imagery from Denmark, from Tue Hassenkamp's laboratory, published last year, showing rings directly under AFM, visual sort of verification that we're forming RNA, because single nucleotides can't form rings. So this was done under the conditions of the AFM imager itself. So rings, viroid-sized rings of RNA. So all of this evidence in the laboratory suggested that if we went out to hot springs, we could introduce these chemical compounds and form polymers. We also need to form stable lipid vesicles. So we went to Yellowstone National Park a few years ago to a hot spring like this one, which is silicious and alkaline. And we took samples of water from this hot spring and from an acidic hot spring and introduced amphiphilic compounds that form membranes. And you can even see in this vial that I'm holding up that it's a little cloudy, and that's because there's a combination of compartments, micelles, membranes in there. Placing it under a microscope, if we do wet-dry cycling, we can encapsulate from these membranes formed in hot spring waters, we can encapsulate DNA as seen by the fluorescence that you see in these compartments. When we take the same compounds that form membranes and we place them into seawater, they crystallize, they kind of clump together, and they can't form stable compartments. So this is another demonstration of why you need fresh water at the origins of life from these simpler amphiphilic compounds that can form stable compartments. So we decided to try to do the polymerization. We formed the compartments. Now we have to form the polymers to go in the compartments. And this is at Hell's Gate in Rotorua, New Zealand in June 2018. We placed a heat block down of our vials of our monomers. And then I simply cycled them with a pipetter with acidic and alkaline hot waters. And the acidic water works because it protonates the solution and you can get those ester bonds forming. The alkaline water we don't think can really promote much polymerization, and we use it almost as a control every time. And what I want to point out here is this little vial where you can see just these films there, and those are the layers of lipid that have dried down, and between those layers are the monomers of RNA, kind of getting baked and moving around. They have mobility, and as water leaves, they click together into the polymers. And so the objective is to form these in the hydrothermal conditions of the hot spring, and we were able to do this. As you can see in the laboratory, we have these little smudges here that indicate a certain length of polymer formed, and in the hot spring condition, we're forming polymers above the 200 mer, the 200 chain length basis. Tends to work best with mixtures of AMP and UMP. So again, when you form these compartments with polymers in them, you create almost a kind of roulette wheel where each little compartment that has polymers present is a natural experiment. They're like chips on this roulette wheel. And as you turn it, by their trillions, functions can emerge because you're subjecting these little compartments to stresses. And you can get a sort of pre-Darwinian selection going, a kind of combinatorial selection. So let's take a look. So we have wet, dry, and moist, and you can see the dry phase, that little film forming the polymers. When you put some solution in, you form these protocells bud off. They're tested for stability, as you can see in this case. The ones that are stable then return into this gel phase we call, it could be like a progenote, as Carl Woese suggested. And then the whole cycle starts again, wet, dry, and moist, wet, dry, and moist. And so the dehydration is here. There's a freeze fracture showing those layers. Here's budding of protocells off. And here's the delivery of the surviving protocells that are still stable, still holding their compartments together and with their polymers. And then the polymers can be subject to elongation or to further complex changes, just as Charles Darwin was calling for 150 years ago. So the whole system is a kinetic trap that cycles these populations and can give rise to what we call the progenote, a transitional unit carrying simple protocells to complex living microbial cells, a sort of engine of creation or complexification, if you will. So this engine turns through these cycles, and what is predicted, and this is where our science stands, that polymers will emerge that could have specific functions that are selected through combinatorial chemistry to stabilize their compartments, to perhaps do other functions that are selected through this entirely pre-Darwinian process, because there's no genes, there's no templates, there's no even quasi-genes in the beginning. So this is kind of the model that we have, where the challenge that we remains for the next decades is to run these experiments and look for functions. So let's take a look at the what we mean by functions. Let's go to a nice cartoon model of a factory that makes tools by chance. You could have such a factory that would make completely any shape of tool you want, but these tools don't necessarily do anything. So how do we get from tools that are just simply made by chance, in this case, polymers with unknown functions, to tools that do jobs? It's all done through this wonderful process called selection. So let's take a first tool, the needle. Well, what would a needle do in a polymer world? And these are just metaphors. But what if this needle stitched the membranes together so they were more robust? So this is sort of the metaphor of we're budding off our little protocells. They have this needle function that makes the cell wall more stable, kind of like patching your bicycle tire to keep it from deflating. Well, you'll get more needles in the system. So what if the needle was joined by a new function, a little corkscrew, which made a hole in the protocell? Well, that hole might be important because it would let certain molecules in and other ones out. And maybe ones that create osmotic stress are let out so the bubble doesn't pop. And maybe the same hole lets nutritive molecules, little solutes, in, such that when an energy capturing function or tool, such as this magnifying glass, emerges, then you can start something called metabolic cycles to make things. So when we get all these tools emerging, all kinds of things can start happening. But the main thing is that protocell becomes more persistent, more robust. So one of the challenges is, how do you make the tools themselves? Well, you need some kind of mechanism, and here's our little Swiss Army knife, to collect these tools together into a way that they can get copied between each other. So for example, here's a little Swiss Army knife representing perhaps a quasi-gene, the first templating system that can code for these tools and make perhaps little peptides and RNA. So it's a proto DNA that can make sets of these particular functions, which might be very short peptides. So once you can do this, once you can make copies of these sets of functional polymers, you have the opportunity to have another tool arise, which is the little scissors here, which can snip things apart. And this snipping tool is your first fission, the first way you can divide sets of tool-making tools. And the division of these protocells into daughter compartments would enable the transition to the living world. So as soon as you're able to take the protocell, it divides, it has the building blocks it needs for the next protocell to emerge that is functional, that can then divide after that. Then you have the transition to the cellular living world. And this is our prediction that in the decades to come, colleagues will run experiments, maybe with automated combinatorial chemical systems, to show the emergence of functional polymers from stabilizing polymers that simply stabilize the protocell, to forming pores, the emergence of metabolic cycles, the emergence of a replicator and catalysts that can catalyze their own replication, all controlled by feedback polymers. So all this has to emerge on its own through cycling systems up to the point where you have protocell division into living cells. So each of the functions has to begin, and we predict that it's going to happen in this horizontal sharing aggregate proposed by Carl Woese decades ago as the progenote. It's not done through strict competition through individuals. It's all through, in a sense, a horizontal gene transfer. And I know that Sohan has really focused a lot on this in his previous meetings, that horizontal gene transfer was dominant at life's origins. So just wrapping it up a little bit here, the scientific and public interest in the hypothesis has been growing. This was a cover article in 2017 in Scientific American they produced this nice visualization, this little spiral diagram of the wet-dry cycling driving complexity. Astrobiology published the article last year, and it's still very widely read. The statistics show that I think it's currently the fourth most read article in the journal. And then Nature, about a year and a half ago, published a story that was quite controversial on the transition of our colleagues in the field thinking from the oceans, from the hydrothermal vent approach to the origin of life to an origin of life in freshwater environments on land. So this was an article that was kind of part of this paradigm shift that's undergoing in our field. So let's look at the implications. The implications are fairly profound for our field, for our colleagues, in that recently it's sort of been seen that there are two alternative hypotheses for life's beginning. One is that hydrothermal vents of the ocean, as you can see in the bottom here, that life emerged from fundamental building blocks synthesized in the vent environment from basically fixing carbon, using the gradients, using the energy in these environments, and that all the way up to the building of polymers, to the creation of compartments, selection, et cetera, that life could emerge in such an environment and then adapt to the marine surface and start capturing sunlight and then emerge on land, and that you carry out, so it also emerge in the ocean and emerge on land as marine algae and then to land plants. But this is a very challenging environment. This is thermodynamically uphill, as identified by Steve Benner. It has the water problem. Anything that's made at the vent environment is subject to hydrolysis and can break down rather quickly. So this is called the water problem or the water paradox. So the challenges here in the environment are quite substantial. That it has not been demonstrated chemically, experimentally in the last 30 years, a plausible path to form these monomers, concentrate them enough to form polymers. And there's no mechanism for selective cycles for this pre-Darwinian selection that's needed. Whereas life emerging in pools on land, this has been experimentally demonstrated up to the point that I just discussed. So it seems to us that it's more plausible that life emerged on the land, adapted to the seashore, but continued to inhabit the land to the origin of freshwater plants, which have now been shown to have originated in a freshwater single-celled organism, is the ancestor of all land plants. So it's a completely new kind of concept. So really, land-based scenario is thermodynamically more plausible and more supported by experiment at this point, including fossil evidence in the rock record. So the second implication is where we look for life on other worlds. So on Mars, the Spirit rover discovered opaline silica, and you can see this little trench dug by a failed wheel. This is about 15 years ago. And this suggested that at Columbia Hills, at home plate, there was a hot spring, very much like a Yellowstone, an ancient Yellowstone. So the Perseverance rover, we proposed with a group, our team, our Columbia Hills landing site team, proposed to go back to Columbia Hills and sample these rocks, which look like digitate opaline silica, similar to what you find in El Tatio in Chile, that is completely influenced and has biopreservation of microbes. So this is an excellent potential biopreservation site on Mars. We did not prevail, and Perseverance landed in Jezero Crater earlier this year. But that site still is very much of astrobiological interest. So I'm going to propose that if you look at life, say on exoplanets, say around a red dwarf for example, shown sort of depicted in an artist's conception here, that to start any form of life requires polymers with encapsulation that then becomes subject to combinatorial selection. And it does suggest that aqueous carbon, with all the basic elements that we see in life on Earth, in cycling pools on subaerial volcanic landscapes, can support this. This is experimentally demonstrable, and that perhaps you need a world that has subaerial landscapes, that has volcanism, that has all these components to be what we might term as urable, able to support the beginning of life. So habitable worlds are perhaps a little different than what we might term as this new term that we're putting forth in the community, urability, which is the ability to start life. Habitability would be the ability to support life that was introduced from somewhere else. So if we look therefore at ocean worlds such as Enceladus, since they have no wet-dry cycling, no volcanism at the surface, no access to exogenous organics, there's no path to polymers or protocells, and so life cannot start at Enceladus, but they still might be habitable. There may still be microbes in the oceans or at hydrothermal vents on Enceladus, yet to be determined. So it's not perhaps an urable world, but it certainly could be a habitable world. And so summary, life began in freshwater hydrothermal pools, subject to wet-dry cycling. And that this is a wonderful statement by Dave Deemer. Life did not invent nucleic acids. Instead, the chemical energy made available wet-dry cycling assembled random RNA-like polymers. And that if amphiphiles or membrane-forming molecules are present, then the polymers can be encapsulated in these vast numbers of compartments called protocells, each being in a microscopic experiment. And then, of course, protocell populations can undergo selection and evolution. So this is the final statement. The prediction to be tested is life began when selected systems of encapsulated polymers emerged that were capable of catalyzed metabolism, growth, and replication. A good definition for an origin of life. And finally, the new experimental approaches. Here's what we challenge ourselves in our field to test in the future, that when you get aggregates of protocells together, they form a network. And there's some exciting work going on in Australia, in Norway, in Irene Chen's group, on protocell to protocell diffusion, stability, the idea that the network effect within protocell aggregates or progenotes is very important in life's beginning, because it's not just polymers within a single compartment. So that these represent the Carl Woese's idea of the progenote, and that you could see that a hierarchy of reaction networks can emerge within these systems. And we're also predicting that much of the chemistry, and this is something for Sohan and your group to consider, much of the chemistry happens co-located on membranes. That the volumes of these protocells, which are cell-sized compartments, are just too dilute, too disorganized to support much chemistry. But if polymers are attached to membranes, they're going to be traveling around and encountering other polymers and solutes, and creating transient pores, for example, so that perhaps most prebiotic chemistry is co-located on the membrane. And this is work with our colleague Roy Black at University of Washington, very exciting direction for the field. And so we're just predicting that, that all of this now is open to experiment in the next decades. You can find the article in Astrobiology, it's open access, just Google hot spring hypothesis for an origin of life. And just closing with the acknowledgements and a little special note, thank you for Sohan and Norcell and everyone here. These are our main collaborators. And our Biota Institute, as you can see the logo in the lower right, we're gathering support and mentoring young investigators, students to postdocs who are entering into the field of astrobiology, specifically interested in the origin of life and its implications. So check out biota.org. That's where you can find a lot of really good resources, videos, documentaries, articles on the hot spring hypothesis.